All right, so in this video, I'm going to be going over the link reaction as well as the citric acid cycle. So if you didn't watch the last video, we have glucose, which was 6 carbons in length. That is going to be broken down into 2 pyruvates, which are 2 3-carbon molecules, okay? So my best piece of advice for you guys studying this, if you haven't been, is follow the electrons. And now I'm going to tell you to follow the carbons, right? So in glycolysis, you didn't necessarily make any substantial amounts of ATP. You made ATP via substrate level. You did make some NADH. So go back and watch that if you are feeling confused on this at all. But notice we're outside of the mitochondria. Glycolysis takes place in the cytosol. We're going to focus on this video on these 2 steps, pyruvate into acetyl-CoA and the citric acid cycle. So before we do that, let's jump to this little animation I have pulled up here. So these are your 2 pyruvates that you had made from glycolysis. So I just want to make sure that you guys are understanding where they came from. Came from glucose. So each one of these black spheres represents carbon. So each one of these is going to lose some carbon, and it's going to form carbon dioxide. I'm pulling electrons off. I am making that's where your CO2 goes out to the atmosphere, and I make 2 of these acetyl-CoAs, okay? So these are just enzymes that get attached onto these 2 carbon molecules. So this is going to be referred to as what's called the link reaction. So let me kind of just tell you what you need to know about this as simple as possible. So glycolysis to the citric acid cycle, the point of the link reaction really is to make acetyl-CoA. So you don't make ATP, so just understand that this pyruvate down here, really you have 2 of them because glucose makes 2 pyruvates. This is going to be where your CO2 leaves, okay? It's the first time you're seeing CO2 as a byproduct, and that goes to the atmosphere, or it leaves the cell, okay? So that CO2 leaves, goes to the atmosphere. Now, the key thing here is that you make some NADH. So NADH is eventually going to go to the electron transport chain. So even though you don't make any ATP, you do make some NADH, which will eventually make ATP. So a couple of things here. It takes place in the mitochondrial matrix. So we're actually inside the mitochondria when this takes place. So pyruvate goes in, you make your acetyl-CoA. Really, this coenzyme A is just this big old enzyme that gets attached on it. So that's really going to be your link reaction. No ATP, but you do lose some CO2. So you've actually lost CO2 from glucose. So let me show you this clip real quick. So this we'll just revisit where pyruvate comes from. So acetyl-CoA formation. I like this video because it's showing you there's your 3-carbon molecule. You're in between the 2 membranes here. And this 3-carbon molecule, right, this pyruvate, I'm going to lose some CO2. So that carbon leaves. This is your 2-carbon molecule. I'm going to lose the electrons. I make NADH. And then here's that big old enzyme. And now this we can refer to this as acetyl-CoA. All right. So that is going to be your link reaction. So moving forward, let's kind of talk about the next step involved in all of this, which is going to be the citric acid cycle. So moving forward, let's talk about it. This is just kind of a revisitation of the link reaction. But let's look at the citric acid cycle, and I will go over this first. So we made your 2 acetyl-CoAs. Let's see what happens next. So now we're actually inside the mitochondria over here. There's this 4-carbon molecule called oxaloacetate. And then notice what happens. That acetyl-CoA you made is going to combine with these 4-carbon molecule that was already there. And now you've made what's called citrate. Now the point here is that this has a lot of electrons to give off, okay? So in terms of this, you're actually going to be losing some more CO2 that goes out to the atmosphere. I'm going to oxidize this, and I'm going to reduce NAD to NADH, lose some more CO2. I make more NADH. I make a little bit of ATP via substrate level phosphorylation. Again, I've made some FADH2, which is basically the same thing in terms of functionality as NADH is carrying electrons. But notice what happened. I remade that oxaloacetate, and hence the name cycle. 1 turn, 1 pyruvate made 1 acetyl-CoA, 1 acetyl-CoA combined with 1 oxaloacetate. So a really common question is that if you had one glucose molecule, how many turns of the citric acid cycle would you get? You would actually get 2. So let's look at the next one and we'll play this out again. So I've already made this from one glucose. I'm going to take that other acetyl-CoA. Again, this came from glucose, combines with that 4-carbon molecule. I'm going to be pulling off some CO2. But the key thing is that I'm actually making, look at all this concentration of NADH I'm building, okay? Yes, I make a little bit of ATP here via substrate level phosphorylation. And this is basically what happens in the citric acid cycle. So let's get into this real quick. I want to emphasize a couple of things here. What you should really understand bigger picture. Really, what is the point of the citric acid cycle? And it's to make NADH / FADH2. Okay. Now this is just showing you for 2 turns, right? Because you make 2 acetyl-CoAs. Keep track of where that came from. Well, that came from pyruvate. And where did pyruvate come from? Well, that came from glucose. So glucose makes pyruvate, pyruvate makes acetyl-CoA. So I should be clear here. 1 glucose makes 2 pyruvates, 2 pyruvates makes 2 acetyl-CoAs. But again, the point is you're making NADH and FADH2 for the electron transport chain. So yes, you make ATP. Some books, and a more appropriate term is you actually make GTP here. But functionality is going to be the same. And basically this just goes to show that you always remake this 4-carbon molecule. That way, every time you get this 2-carbon intermediate of acetyl-CoA, you can remake this citric or citrate molecule, okay? Now, this is from the POGL, and this is just really good to show you that citrate has high, very high potential energy because look, you remake this really high energy molecule every time. Oxaloacetate is there, so it's that 4-carbon molecule, so I'll just go ahead and circle all the carbons, 3, 4. Again, acetyl-CoA has 2 carbons, okay? So the 2 plus 4 is going to give you this 6-carbon intermediate. 2, 3, 4, 5, 6. It has a lot of potential energy. A lot of electrons can be given off, okay? So don't fixate so much on all the names. You're not necessarily required to memorize them. Key thing here is you lose some CO2. You make a high amount of NADH and FADH2. Again, what is FADH2? It's just another electron carrier. So let me show you the other visual here. So we will, let me just zoom out real quick. If you go to this clip, let me replay this. This is showing you your acetyl-CoA. There's your big enzyme. This is your 4-carbon oxaloacetate molecule. So when you let this play, notice these combine, the enzyme leaves. This is now citrate. Watch what happens here. So this citrate has 6 carbons to give. I shouldn't say 6 carbons to give, but it has a lot of electrons. I lose some CO2. Look, there's NAD. Picking up the electrons is going to become NADH. Okay. Lose some more CO2. I'm going to lose some more electrons. This is going to randomly form some ATP via substrate level phosphorylation. That's just showing you that another one happened at the same time. So there's some ATP or GTP via substrate level phosphorylation. Look at this one's in green. That's FAD. Picking up electrons becomes FADH2. This is going to pick up the electrons even more, making more NADH. So really, you reform oxaloacetate, and look at the whole process starts again. So key components here, citric acid cycle, what's the point? Really, it's to make a lot of NADs and FADs. And again, think backwards of where it all happened. Pyruvate formed acetyl-CoA. Pyruvate initially came from glucose. So a really nice overview again. So that is the citric acid cycle. Next video, we're going to talk about where does the NADH and FADH go? What is the point of making all that?